Awesome. So this is a little bit of a different talk than Luke. I, I have, it seems like so many fewer negative experiences with being scooped and being in all sorts of different ways abused by the review process that I just feel like, you know, it makes me very thankful to hear these stories that it could be so much worse. So it's so nice. It's so nice to see that he's still smiling about it all and still laughing. Though I also feel a little bit of the vicarious pain. Okay, so so far today, a lot of the talks have been about imaging data. So this is going to be a slight change of pace, talking about data, so spike data recorded using extracellular electrophysiology. I wanted to sort of start out with like a big picture question about decoding. So why do we do decoding? And Luke's reason was somewhere here, I think. So trying to understand something about the brain. So when I entered this field, I started up here at the top, thinking about using brain activity to control some part of the external world. That there's been a lot of work in that area. I'm going to show you some videos in a little bit. Very recently, I became aware of the fact that Facebook's like top secret organization is doing some sort of brain decoding stuff these days. Also, and in their case, their goal is to try to reconstruct speech, right? So that you could, I guess, so that you could control your phone with your brain, because what we all really need is the ability to type Instagram messages faster. Big picture, right? The idea is we have stuff in our brain. We might want to get it out in order to control something in the world. So if we're paralyzed or have some problem that blocks that control, then we might want to be able to restore it, right? So that's sort of the therapeutic reason to do decoding. Another parallel and not necessarily completely orthogonal reason is because we want to understand something about what's going on. So by testing a decoding model, we can say something about the encoding model. So we can say something about how information is being represented. And if something is changing in the way that that's being represented, then we can learn something about the dynamics of different brain circuits. And the final and most interesting thing, I think, is when we're talking about brain states that are not directly observed, where we actually have no, the thing that we're decoding, we have no way of actually verifying whether what we're decoding is true or not, so internal brain states. And I think that's what's most exciting. In the case of fMRI, you guys have a really neat opportunity over the whole brain. In the case of spikes, often it's much more challenging because we're only looking at a small slice of the brain. So Matt, after I do this little intro, is going to talk about hippocampus and hippocampal replay. Okay, so when you're thinking about decoding algorithms, you should, how many of you are familiar with this idea of supervised versus unsupervised learning? Awesome, okay, cool. So I think almost everybody here so far has been talking about supervised learning. A little bit of the clustering, these sorts of things are unsupervised, but in general, we think about having something that we're trying to match our decoder to, right? So in the case of supervised learning, we're minimizing an error of some sort. I think all of the talks so far have been what I would think of as discrete decoding contexts. So where we're looking at a single snapshot of something and we're trying of neural activity, and we're trying to estimate something that represents. So that single snapshot is something that is divided into a discrete set of possible things that could be decoded. That's as opposed to something where we have a continuous model of the world. So for example, the arm is moving in a continuous space and we want to represent that continuously. Okay, so a lot of the algorithms that I've heard named, I would think of as being non-probabilistic classification approaches. So these are things where we can use the fact that we're doing supervised learning to minimize error. So support vector machines, even things like principal components analysis can be thought of as non-probabilistic. My favorite way of approaching any decoding problem is to start with a probabilistic model, because that means that I can write something down. And I'm going to show you what I mean by that in a second. So yeah, let's just go on to that second right now. Okay. So how many of you have seen a graphical model before? Okay, most everybody, good. So here the idea is that this Z is this thing that we're trying to decode, right? And the thing that we're observing are the activity of these neurons. And so here this one is time, so this is at time step 1, but whatever. I think of neural signals as evolving over time, so time is always going to be a variable here, right? So when I write this graphical model down, what this means is that I believe that the neural activity is probabilistic, meaning that I can describe it as with a probability distribution, and that probability distribution will be dependent, will have a dependency on this the variable that I'm trying to decode, right? So in this case, if this is the arm position and these are the neural activity, then I would say that the neuron 1, neuron 2, neuron 3, neuron 4, neuron N all depend in some way on the arm position. And this, I can usually, I assume that I can write down what this probability distribution is. So often it's going to end up being Poisson, but I'll talk about some other models in a second. Okay, so this is the way that we often think about things in the case of the hippocampus. So each time slice we take as being independent. In the case of motor processes or things where the variable that we're decoding is changing across time, we'll have a different model, which I'll show you in a second. Okay, so I mentioned continuous versus discrete. So in the continuous case, this probability distribution is a function of the variable that we're decoding. And that means that when we invert it, this is going to be a little bit hard. In the case of discrete variables, what we're going to say is that the variable that we want to decode can have some set of values. This morning, for example, there was 4 different kinds of picture, right? So we want to decode that this is that the brain is representing an insect or I forget what a bird, right? And then which kind of insect or which kind of bird. And so then we would say that the neural activity was going to have a different probability depending on which of those 4 classes it was. So these discrete models are much easier to deal with, but these continuous models allow us. So obviously, if we want to classify all insects or all images, then it's hard to have a discrete model of the world in the case of where our actual space is something that's continuous, right? So in those cases, we want to have something that is a probability distribution that continuously varies. So, okay, when we do decoding, what are we going to do? So we start here at our graphical model, and essentially what the simplest, naivest way of doing decoding is to invert this probability distribution using Bayes' rule. So Matt will talk about that. And in the case of these discrete probability densities, that's very easy because the normalizing factor is a sum. In the case of the continuous random variables, it's more complicated. So I will now add one more possibility, which is now if we have dynamics, which means that the thing that we're decoding is changing across time, then now stuff becomes more complicated, but also becomes better in a sense, right? So if I know that if I'm a rat and my hippocampus is representing where I am now, then my expectation is that in the next time moment, my hippocampus better not represent that I'm over by the coffee, because that would mean that I'd teleported, right? So you can think of these arrows as representing the dependence on the fact that my behavior can't involve teleportation. In the case of arm movements, for example, if my arm is here now, it can't be behind my back in the next instant because it takes time, it takes effort for me to move it around. So when you have this kind of model that relates the thing that's being decoded across time, then the appropriate decoding approach is a filter. And so often we'll think of these as Kalman filters or particle filters, or depending on what the actual distributions are. So we have this the probability distribution that describes our neural activity dependent on our variable that we're decoding. The most common thing is actually a Gaussian. So almost every approach that you guys use where you don't think about probabilistic things, probably in the end it works because you're assuming that your data are Gaussian or close. When we actually want to assign a probability distribution to our neural activity, we often will pick a Poisson process because for spiking, Poisson processes make sense, right? Spikes come in discrete chunks, and so it makes sense that we should have a Poisson process. Turns out if you smooth your data, as Matt likes to do, you sort of break your Poisson assumption, and the statistics are not quite perfect anymore. But empirically, smoothing actually makes things work a lot better, so we still do it. If you want to be more fancy and you're dealing with spiking data, then you may want to think about refractory periods. You may want to think about bursting. And you can model all of those statistically, and there's some good work on that. My experience is that doesn't really improve decoding that much. OK, so when we're doing decoding now, historically, the most common thing to do was a population vector. So what is a population vector? I'll show you an example in a second, but a population vector basically says, let's pick something that we'll call the preferred direction, which characterizes every neuron, and then scale that preferred direction by the number of spikes that the neuron produces, and then add all of those up. This is not probabilistic. It's just sort of an empirical thing, but it worked quite well for some early motor cortical stuff. The Bayesian, which I call independent Bayesian here, meaning that this is independent across from one time step to the next, inverts this probability distribution. Then there's the recursive Bayesian approaches, where we have the thing that we're decoding is connected across time, so the Kalman filter, the particle filters. And then people that do continuous decoding for spikes these days for prosthetic purposes, where they're really trying to get data out in order to control something. The most common thing that appears to be done nowadays is to project into a lower dimensional space, so find a lower dimensional space that represents the neural activity, and then to decode from that lower dimensional space. Is there anybody here who's doing anything like that? Who's doing like motor decoding prosthetic stuff? No, that's too bad. Okay, cool. Yeah, it's a fun space, but it sadly is limited by the fact that it's unclear what the final outcome is going to be, I think. Okay, so cool. So here's the first step, right? So here's spikes from a single neuron in motor cortex, right? And so we see that if we look as an animal makes center out reaches, that there's a direction in which the neurons produce the most spikes. So I think in this case, it looks like it's somewhere around here for this particular neuron. So these are rasters. You guys are familiar with spike trains, right? So these are rasters across trials. And there's actually something interesting here, which is that you can also see that there's an increase and then a decrease around the time of movement. Why do the spikes start firing before the arm starts moving? Well, because they're driving the muscles, right? This is causal, right? So we see something where there's an increase right before the time movement, and then as the movement goes, and then stops. Okay, so why is there an increase in spiking in the contralateral direction when the movement stops? Anybody? Basic neuroscience. Sorry, let's pause. Let's ask that as a question just to make sure you guys are paying attention. Okay. So this is the preferred direction. This is not the preferred direction. So monkey's moving his arm in this direction. We get many spikes, then few spikes. When the monkey's moving his arm in this direction, we get few spikes, and then extra spiking here. So why is there a slight increase in firing? And yes.
Because there's some sort of lateral inhibition between the preferred direction and the non-preferred direction, such that when the other part decreases, the non-preferred direction gets reduced from inhibition.
That's a good answer. That's not the answer I'm looking for. Any other thoughts? Yeah, that's the answer I was looking for. Is that it's the in order to accelerate, you move your muscle. In order to decelerate, you have to move the antagonist muscle, right? So when you're moving to stop, you have to. Has anybody, have any of you ever been asked to give directions on how to change lanes in a car? Okay, so this is a very random aside. You, friendly person who answered this question, I want you to give me the exact steps about how to change lanes in a car.
All right. First, you look in the rearview mirror.
Yes.
Then you look on your right.
Okay, good.
And then if there's nobody,
Yeah.
you just accelerate.
Uh-huh.
And then change lanes.
But tell me what you do with the steering wheel.
I turn it towards left.
Okay, good. Uh-huh. And then what?
And then I stay on the lane.
Uh-huh.
And then I pass the car. If
Uh-huh.
I pass the car, right?
Uh-huh. Uh-huh.
And then I turn. Well, and then I check if there's another car coming.
So this is very interesting, but you did exactly what I did. So when you next time you get into your car and you change lanes, watch. So when you go into the new lane, you turn like this, just like you said, but then you actually have to straighten back out. So you actually have to turn your wheel back in the other direction, right? So sorry, so that was a relevant thing, right? So the muscle, the contralateral muscle has to be activated in order to stop the arm from moving. Which is interesting if you're trying to decode this in the context of continuous movements as opposed to discrete movements. So this is data from I can't say his first name, Georgopoulos, who was one of the first people to record a motor cortex in monkeys during arm movements. And he used the population vector, which is a very simple way of looking at these data. I've already described this, but essentially what he was faced with was a very decoding problem. What is the decoding problem? The decoding problem is that you have many neurons and you want to combine their information. What I described was ways to do this probabilistically. So the Bayesian approach that Matt's going to talk about is a probabilistic way of combining the activity from different neurons. The population vector is a very naive way of combining the data across multiple neurons. But what it does is it actually works quite well. So it says, because we know that these motor cortex neurons have a preferred direction, we can pick that preferred direction and assign it to each neuron. And then whenever we see a novel movement, we'll take all of the spikes from all of the neurons, weight their preferred directions by that value, and then add that, do a vector sum of those weighted population vectors. And what we get is we get something where the arrows point in largely in the right directions. So what was cool is that he was able to take this population vector in a task where a monkey had to now make a mental rotation of what he was supposed to do. So he's given a cue of where he's supposed to move, but that cue was shifted 90 degrees from, the cue was 90 degrees from where he was actually supposed to move. And so what he saw is that in these monkeys in motor cortex, that the population vector initially pointed in the direction of the cue, and then it shifted to the direction that the animal was supposed to move. Okay. So this is sort of the early versions of these, and then this is now some more recent data. This is from the Schwartz lab from probably the early 2000s. So this is a Kalman filter being applied to neural data from motor cortex, controlling, I think, just 2 degrees of freedom of this robotic arm. There's a monkey who's feeding himself, and this is from my old lab at Stanford. This is discrete decoding. So now in this case, the animal is supposed to, sorry, there's no sound. Hold on, the sound here is fun. Oh, that's too bad.
You put the mic down by the computer.
That's okay, it doesn't really matter. So let me just quickly pause this so you can see what you're seeing. Okay, so what happens is that these are reach tasks again, except it's on a touch screen. And the animal is supposed to reach to this target, except in this case that there's online decoding going on. That's discretely decoding which of 8 targets the monkey is intending to move towards. And if he makes a mistake in this, in planning to move to the right target or to the wrong target, then he's forced to actually move. And at the end of a chain of these, he's also forced to move. So what you'll see is that he's properly moving to 5 targets, and then he makes a movement, and then he moves to another 5 targets, and then he makes another movement. And so there's actually the target that he's planning to move to is being decoded here in these 200 millisecond blocks. Okay, So I'm going to stop there. Matt's going to come and talk about decoding in the hippocampus in much more detail. I'm going to skip over this unsupervised learning example. But do you have any questions sort of about the sort of big overview of decoding of spikes before we do that? No? Okay, awesome.
Yeah, so I don't plan to spend a lot of time on this, but I'd like to give you a little bit of a worked example of how you would actually, in practice, implement one of these decoders and what are some of the practical issues that come up. So I've shown you earlier this type of plot where you have an animal moving along a track, he's moving in space, and you get these neurons that are selective for particular locations along that track, the place fields, and so you can order them such that, well, you see this smooth progression from first you activate the place field over here, you keep walking, you activate a neuron in the place field over here, so you see this sort of diagonal showing up in a raster plot. So what we can do is cut up the neural activity here in discrete time windows and sort of schematize it like this, where you have the activity of, say, 2 example cells. And you may be blind to the rat's actual location, but you can ask, how well can we guess or decode the actual location of the animal given that spiking activity? And the way this is commonly done, which Caleb introduced sort of as this probabilistic Bayesian decoding approach, is that you already have some information about where these different place cells tend to be active, and you can formalize that in the form of a tuning curve, where you just say, what is the average firing rate for all these different locations along the space that the animal is moving through, the stimulus space. So you can forget about these labels here. This is just position. So here you have a cell that maybe is active in 2 different locations. Here you have a cell that sort of has a much broader tuning. You can imagine you have a whole populations of these things that you're observing some spike counts for in some given time window. And sort of the intuition for this is relatively simple. You can just think, well, if you observe 3 spikes from cell 1, and on average this is cell 1's activity, you would probably say, well, it's much more likely that the animal was in these locations where on average there's a non-zero firing rate. Otherwise it would be extremely unlikely that you saw 3 spikes given a very low average. Likewise, if you saw 0 spikes in cell 2, and you know that on average cell 2 is sort of active in this sort of way, you'd say, well, 0 spikes is more likely to occur when the animal is over here than when the animal is over there. So you can formalize that mathematically by having some assumption about how the possible spike counts that you might observe are distributed. So a simple way to do this, as Caleb indicated, was to assume that these spikes are distributed according to the Poisson distribution, which just says, given some mean firing rate, what is the probability of observing various other firing rates around that mean? So what we're really doing is saying we're trying to estimate, what is the location of the animal, the probability of location X, X1, X2, and so forth, given a bunch of spike counts that we're observing. So if we apply Bayes' rule, we would just want to invert that to say, well, we can compute this by knowing what is the probability of observing some number of spikes given that the animal is actually at specific location. So that's this thing here of saying, well, for each of these locations, we can say what is the probability of having observed the number of spikes that we did. And basically the details of the equation here are just given by the mathematical description of these Poisson distributions. Like it tells you what is the probability of observing 2 spikes given that your mean was 5, the probability of observing 3 spikes given that your mean is 5, and so on. So you can just iteratively go through all these possible locations and use that equation to find out how likely was it that the observed number of spikes was generated by a mean of some number, right? So then for each cell, you get this sort of posterior, and you have a whole population of cells. So again, one sort of the stupidest way of obtaining a population decoded estimate is just to assume that all those cells give you independent information. You just multiply all these probability distributions together to get sort of a final estimate. And then what you can do is say, well, I actually can look up the actual location of the animal in this case and have some sort of error metric of, well, how well did this correspond to the animal's true location? And you can compare that for various assumptions about how your spike counts are distributed or various other decoders. So I think this is really the central idea of how a lot of spike-based decoding ends up working in practice. So there's sort of these two different steps where you have to first find what are these tuning curves for all your cells. So that often can be thought of as, what is your encoding model, right? How do you go from where the animal is in space to some firing rate? And then you end up inverting that, using Bayes' rule to do your decoding. And then you can use that to do a variety of interesting things. So I don't want to go over time too much, but just to give you an intuition about some of the categories of things you can do that Caleb mentioned. So you could say, well, how well can you decode, say, a variable such as location from neural activity in different brain areas? And you might think, well, hippocampus, everybody talks about it as having place cells and Tolman's cognitive map and so on. We should be able to decode location really well. But in this particular study, we found that if you compare the spatial decoding accuracy, which is here on this axis, between a number of different brain areas, that actually in dorsolateral striatum, in that particular task, we could decode location better than from hippocampus. Again, there's some interesting explanations underlying that, and you can ask how well it generalizes and so on. But this is just one sort of example of how you might apply a decoding method just to make some comparison between different brain areas. And the other application that I'm particularly excited about is this idea of what about these internally generated brain states, right? Where you have a situation where you don't really have a ground truth to compare something to, but you're thinking about memory, about imagination, about predictions, where you can apply a decoder that you've sort of trained on the situation where you know the correct answer, and now say, even though the animal is no longer running, but maybe having a memory of some earlier run, can you just apply that same decoder? And this is sort of I'm going to skip a whole bunch of slides, sort of the idea of replay, right? That now in an individual episode where the rat isn't actually moving, you run your same decoder and you look at the output of that, and the sort of things you can find is that, for instance, on a T-maze where you know that the animals only ever run left from the choice point or run right from the choice point, so just that image in the top left, you can now look, okay, in this particular case, where does an individual replay map onto space? So you can see that the animal replays this experience forward as well as backward. So what the decoder now allows you to do is say, even though the animal isn't actually moving, we can map that neural activity onto this track and look at some interesting features of that decoded representation. So you not only get literal replay, but you get a backwards replay, and you might even get a shortcut, a sequence that combines experiences that the animals had separately into one combined episode. So in the case at the top left, there's a replay sequence that goes across the top of the maze, even though the animal has only ever gone left or right from the choice point, but never run across the whole thing. So it's an example of using decoding to access something that is purely internally generated, rather than just saying, how well can our decoder recover something that is known. So just to give you one more example of that idea. So here's a situation where the animal is running around in a sort of open field, sort of 2 meters by 2 meters. There's all these possible reward locations. And as part of this task, the animal frequently has to return to a home base. Now, before the animal makes such a return journey, you can again decode these internally generated sequences of activity and say, well, under a hypothesis that you think that the hippocampus is about memory, then that activity should kind of replicate where the animal came from. But if you think it's more about planning, about finding how you get to a goal, then those sequences should be more about a trajectory towards the goal location. And this is exactly what Brett Pfeiffer and David Foster found, that if you align all these decoded trajectories to the goal direction, which is sort of directly upwards from the midpoint. So the rat is here, he's about to start a trajectory towards the goal. Now you just line up all these decoded replays. You see that they have a clear bias towards the goal rather than a bias sort of where the rat came from. Now there's a whole bunch of statistical issues with this, of how you do cross-validation, how you prevent overfitting, a bunch of other things that I probably won't go into now. But the tutorials this afternoon for the spike section, I just want to highlight in the tutorial channel in Slack, there are links to the data sets. So depending on whether you'd like to do this in MATLAB or in Python, they actually use different data sets. So take a look at that tutorial channel, make sure that you download the data file before the tutorial starts. But basically, we'll take you step by step through running this kind of decoder, first applying it to a case where the animal's actually running. You can check that your decoder gives the correct output, and then apply it to some internally generated activity and see what sort of dynamics and patterns that you find there. So I'll stop here, and you'll get to explore this stuff yourself later. Enjoy your lunch.